everyone hear me? Everyone in the back? Okay. So yeah, you may be detecting a theme with these 3 talks here on evolvability. So I'll just get started. So I want everyone to think back to the origin of life, and very specifically the origin of self-replication. So as far as we know, our best model for this is the RNA world, where you have self-replicating RNA molecules. And once they arrived, again, as you know, we got this nice increasing evolution of different innovations throughout time, where we had self-replicating RNA eventually leading to the first cells, which were probably close to today's bacteria and archaea, eventually leading to a new domain of life, single-celled eukaryotes, and finally multicellular eukaryotes. So the question I'm interested in is, if you have self-replication, in what sense is this type of process inevitable? Not this exact pattern, and maybe not even the creation of multicellular life or even eukaryotes, but does self-replication mean that you also will get increasing rates of fitness and increasing rates of the evolution of novel phenotypic innovations? So first I want to say what I'm not saying. I'm not merely arguing that does self-replication imply Darwinian evolution. This actually does follow straight from the definition of Darwinian evolution, where self-replication provides heredity, it provides variation, because all self-replication will be error-prone due to the laws of thermodynamics, and it also provides differential fitness. But the main question I'm interested in evolvability is this last one. And the reason is, you could just have a differential fitness where the first self-replicator that emerges is the best self-replicator you can reach. And all the differential fitness would just be deleterious variance. I'm mostly interested in, can you get differential fitness to increase it for beneficial variations? So, I will give the 3rd and 4th definition of evolvability today. So one classic definition is evolvability is an organism's capacity to generate heritable phenotypic variation. However, I'm mostly going to use the ability to increase in fitness through evolution. The reason I'm using this definition is I can easily measure it. And furthermore, I'm going to break it up into 2 different components: optimization and innovation. So what do I mean by optimization? This is what I'm calling the quantitative component of evolvability. And it refers to a fitness increase in an already evolved phenotype. And if you're picturing a fitness landscape, you can picture this as a simple hill climbing procedure. This is sometimes referred as adaptation. However, because evolutionary biologists usually refer to phenotypic innovations as adaptations, I'm going to refer to it as optimization. Furthermore, you have then innovation. And this is the qualitative component of evolvability. And it refers to the evolution of novel phenotypes, such as photosynthesis, or the classic example of a complex feature, the vertebrate eye. And this is not just a simple hill climbing procedure. And as I said, my main question is evolvability inevitable? Or will you get a case like this, where you have a self-replicator emerging, and if this is a fitness landscape, it cannot actually increase in fitness because of the deep valley that occurs here. So how would we test this? So if we're interested in looking at this purely from biological perspective and purely based on how life is on our planet, an experimental RNA system would be ideal for answering this question. However, such systems of self-replicating RNA, and specifically where you can get the emergence of new self-replicating RNA, have not been developed, at least to my knowledge.
That's not true.
Which system? Or we can talk after.
The de novo evolution of the Avida systems is an example. There are experimental systems that do this.
So, okay.
Okay, we hold out the questions.
So to test this question, we need to use digital systems. Specifically, I work with the digital evolution system Avida. In Avida, you have a population of self-replicating computer programs. This is displayed here. They live on a digital grid. Each color represents a different genotype. So as these programs execute themselves, they eventually will copy their genomes. Through copying, they can get mutations, and they'll eventually split off into two separate organisms. Their genomes are composed, each spot in their genome is composed of one of 26 possible computer instructions, and because they are self-replicating, it's an instance of Darwinian evolution, and it allows us to test evolutionary hypotheses in a much simpler system than, say, biological evolution. So one of the benefits of using Avida for stuff to do with the emergence of self-replicators is it has a mechanistic replication process, and it's very non-trivial. It requires the ability to allocate memory to place an offspring, copy its genome into that memory location, and then successfully undergo division. Improving this process is then how I measure the optimization component of evolvability. Also, one other thing with Avida is there are a set of 9 phenotypic traits. This is the ability to perform certain Boolean logic computations. This represents an X NOR gate, and the performance of these computations will result in faster self-replication because in the Avida system, those that replicate faster will outcompete those that replicate slower. And the ability to evolve these traits is the innovation component to evolvability. So just to give you a more visual perspective of what an Avidian is, I'm going to show you a video of an Avidian's life cycle. The first thing you're going to see is it will almost instantaneously allocate the blank genome in which to copy its descendant into. And it just goes and executes the code in itself. As it does that, it will perform certain these logic computations, and then it will begin to copy its genome. As it does this, mutations can be introduced, and that, of course, allows for variation and then selection based on that variation. And after the whole genome gets copied, it undergoes division, and you're left with 2 separate Avidians. So, to study the evolvability of self-replicators, we needed to generate a set of self-replicators. In a previous paper, I studied the spontaneous emergence of self-replicators in Avida. So what I would do is, I would generate 1 billion random genomes for sizes of 8, 15, and 30 instructions. This is just an example of random genomes where each of these letters represent one of the 26 computer instructions. Then what I do, I would test for their ability to self-replicate, because in Avida you can just essentially execute the program and watch what happens. And I also tested for their offspring, at least one of them to be able to self-replicate, because I wanted to avoid situations where it could replicate once and both of the descendants could then not self-replicate. So just to do one of the results from that, which is relevant to this work, so this is the number of self-replicators I found out of the 1 billion genomes. I found 8 self-replicators of length 8, 58 of length 15, and 106 of length 30. Now, it's probably true that self-replicators in Avida are more abundant than they would be in, say, an Arend world scenario, but this gave us plenty of self-replicators to work with to test whether they're evolvable. So now, I then asked, are these randomly generated replicators evolvable? So I did 2 sets of experiments. The first were optimization experiments. So what I did was I created 10 populations for each of those self-replicators of the 3 different lengths. I had 1,000 individuals, and I evolved them for 1,000 generations. They experienced point mutations at a rate of, they had a 10% chance per generation of being a point mutation in their genome, and a 1% chance of getting an insertion or a deletion mutation. And I also set it so they could not evolve new phenotypic traits. This was just focusing on whether they could optimize their genomes for self-replication. So the first result we noticed was unexpected, but most self-replicators initially undergo genome reduction. So this graph, on the x-axis we have all the ancestors, and on the y-axis we have the genome length after 1 generation. And these dotted lines are essentially what their original was. And what happens is, it turns out that many of the self-replicators that arrive in Avida will deterministically copy themselves incorrectly. And what this does cause then is they reduce their genome size to a much smaller length. And it turns out it's actually much more likely to get imperfectly copying self-replicators that emerge than it is to get ones that copy themselves perfectly. The exception is length 8, but that's because 8 is actually a minimal genome size. Yes?
So the one in 15 that ends up at length 8, did you find it back in the group of 8?
Say that again?
The one in the group of 15 that reduces to length 8, that is a member of the other group?
No.
So you
No. These lengths refer to the original lengths. Only.
Yeah, but did you find the one from group?
Oh, did I find it?
Isn't it also
I didn't check, I have to say.
the same one as in the other group?
I don't know. I did not check it. I can tell you that all of them are unique within their original group, but I don't know if it's you're saying essentially it would mutate from this back to one of these.
Well,
Right?
if they're the same.
Yes, I don't believe so, but I did not check that. So the first result then is most self-replicators can increase by fitness by large amounts. So again, I have the different ancestors. On the x-axis, the y-axis is the relative fitness. And what you see is almost all self-replicators are actually double in fitness after 1,000 generations. And they actually can increase by quite dramatic amounts, some of them can. So again, this was averaged over 10 runs, and the error bars are standard deviations. There's one standard deviation, which is why you get that large error bar. So this suggests that in Avida, at least, most self-replicators can increase, are evolvable in terms of fitness. And it holds no matter what based on the genome length. So the next thing I wanted to look at then was the innovation component of evolvability. Again, I did a very similar setup, 10 populations for each self-replicator. However, I only looked at the ancestors which had an original genome length of 15 instructions. I did a population size of 10,000, an experiment length of 10,000 generations. The reason this was it takes longer to evolve phenotypic traits, so I wanted to increase the amount of mutations they experience over the course of the experiment. I had the same mutation rates as the previous set of experiments, and I set it so populations could evolve the 9 phenotypic traits. So what did I find? Most self-replicators can indeed evolve many phenotypic traits. So x-axis is the length 15 ancestors, and the y-axis is the number of phenotypic traits they evolve. This is tasks, so we refer to them as logic tasks. And what you see, all except a couple, evolved almost 2 phenotypic traits, leading all the way up to 5 or 6. And it's actually interesting, this one here. So it turns out what this one is actually the handwritten Avida ancestor that we usually use by default to seed our experiments. And it turns out what does that is it appears to be some sort of trade-off between optimization and innovation. And I won't get why into that occurs. It has to do with how the genome structure is written for it. But it turns out that it's very good at optimizing its replication ability. But on the other hand, this prevents it from apparently evolving novel traits. I will say this is somewhat parameter dependent, though, because this is a lower mutation rate than is typically used in Avida experiments. And it also may be that it's just slower to evolve these innovations. I can't really rule out that it can't evolve them. So the concept of proto-replicators are used in the paper. This was essentially a visual model we come up with to explain what we found. Well, you have where you get the emergence at the beginning of self-replication of a proto-replicator. And it's proto in the sense that no matter what, it actually will not copy itself perfectly. However, eventually it will receive a mutation which makes it stable, and it can, in theory then, copy itself perfectly. And eventually it accumulates further mutations, which then allows evolution to lead to increases of fitness and increases in novel phenotypic traits. So, for my conclusions, almost all Avida self-replicators alter their genome size immediately after replication due to the initial emergence of unstable replicators. Almost all Avida self-replicators are evolvable in regards to both optimization and innovation. And apparently there is a trade-off between optimization and evolvability in terms of innovation that can limit the emergence of complexity. And finally, I do want to say, I don't know how often this will hold in, say, an RNA system, but I do think it's interesting to see that in a system where you do get real evolution, the complexity that the system can reach is usually reachable from randomly generated self-replicators. And I want to end by thanking my co-authors, my PI, Chris Adami, and Dr. Arend Hintze, the NSF Beacon Center for Funding. And I'll take any questions.